Hello everyone, welcome to this video. My name is Zehra Rizvi and in this video I will be explaining y'all few chemical methods of cell disruption. Specifically we'll keep our focus on the use of detergents and alkali in cell disruption, and we will be understanding all this in relation to recovery of microbial fermentation products during the downstream processing. So let us begin with what is cell disruption. So cell disruption is the process of breaking the microbial cell walls and cell membranes to release the intracellular contents of the cell, such as enzymes, nucleic acid, metabolites, etc. Now we all know that microbial cells have many protective barriers like cell walls and cell membranes that maintains the cellular integrity and prevents the leakage of their internal components. Thus, cell disruption simply refers to the use of physical or chemical methods to break down these protective barriers and help in releasing the intracellular contents of the cell. Now, why do we need this cell disruption in downstream processing? First of all, what is downstream processing? So it is the final stage after the fermentation that deals with recovery and purification of the products after the fermentation. Now we all know that microorganisms are used to synthesize many valuable products like enzymes, proteins, various metabolites, etc. Sometimes the product is extracellular, that is, it is secreted out of the cell and it remains in the solution or the broth. However, many a times products remain intracellular, that is, they remain trapped inside the cell. So thus, in order to recover these intracellular products, cell disruption is essential step. So therefore, cell disruption releases the product into the solution or broth, makes them accessible for further downstream purification and increases the overall yield of the product. Now there are several methods which are available, and these are broadly classified into mechanical and non-mechanical methods. Under non-mechanical methods there are physical, chemical and biological methods. As I have already mentioned, we will be understanding only the chemical methods in detail, and in chemical methods the use of detergents and alkali is very prominent, so we will be understanding these two. Okay, so now let us understand our very first chemical method of cell disruption, that is the use of detergents. So first of all, what are detergents? Detergents are surface active agents, also known as surfactants. They have the ability to interact with both water as well as lipids. Because of this property, detergents can very easily dissolve the lipids. And we all know that the cell membranes are mainly made up of lipid bilayer, and as detergents can interact with the lipids, they can very easily disrupt the cell membranes, and thus they are very effective lysis agents. As you can see in the image as well, detergents mainly react with cell membrane of the organism. They destroy the cell membrane, due to which these type of holes and pores are created in the membrane, and from these the intracellular components of the cells are leaked out and ultimately cell ruptures. So this was the basic mode of action of detergents. Let us understand their mode of action in details. So structurally, detergents have two important parts. One is a hydrophobic water repelling tail, that is, it has more affinity for lipids, and other is hydrophilic water loving head, that is, it remains in the aqueous layer, it has more affinity for water. So because of this dual nature, detergents can interact with both lipids, membrane lipids and water. Now when detergents come in contact with the cell membrane of organisms, the hydrophobic tail of the detergent inserts itself into the membrane lipid bilayer. As a result, the normal arrangement of the lipids in the membrane get disrupted, and this disturbance destabilizes the lipid bilayer and increases the membrane's fluidity, which means that the membrane becomes more flexible and less stable, and the lipids in the bilayer, they can very easily or freely move in the lipid bilayer. Ultimately, due to interaction with detergents, the membrane lipids and proteins, they become solubilized as detergent lipids complexes, and the cell loses its integrity and it breaks, and all the cell contents are released in the surrounding environment. As you can see in the image as well, these detergents, they inserts into the lipid bilayer, and the detergent lipids and protein form complexes, and ultimately all the cell contents are leaked out in the environment. About the mode of action of detergents. Now these detergents are classified into three types based on the charges that they have. First is ionic detergents. These detergents carry either a positive charge or a negative charge, and they are considered very strong detergents. A very common example of ionic detergents is SDS, which is sodium dodecyl sulfate. Because of their very strong nature, ionic detergents completely disrupts cell membrane, and they also denatures the proteins, meaning that they destroy the natural structure of the proteins. Because of this reason, ionic detergents are mainly used when protein structure preservation is not required. Second category is non-ionic detergents. These detergents do not carry any charges, and they are milder as compared to ionic detergents. Examples of non-ionic detergents include Triton X 100 and Tween 80. These detergents gently break the cell membrane without causing any severe damage to the proteins. Therefore, they help in preserving the protein structure and its biological activity. Because of this mild nature, non-ionic detergents are widely used for recovery of sensitive fermentation products such as enzymes. Third type is Zwitter ionic detergents. These detergents have both positive and negative charge within the same molecule, therefore the net charge is zero. An example of Zwitter ionic detergents is CHAPS. These detergents cause moderate membrane disruption, and they are less harsh as compared to ionic detergents and causes less protein damage. Therefore, Zwitter ionic detergents provide a balance between effective cell lysis and protein preservation. So now let us understand few fermentation products for which detergent based cell lysis is commonly employed. First, for the recovery of many enzymes, both the membrane bound as well as intracellular enzyme, this detergent lysis is commonly done. For example, in case of pulilinase enzyme, which is a membrane bound enzyme in Klebsiella pneumoniae, detergents like sodium cholate is used for membrane solubilization, and then the enzyme is released out in the environment, which is then further purified. Second, in case of proteins, again both intracellular as well as membrane bound proteins. So for proteins, we saw that non-ionic detergents and Zwitter ionic detergents, they are more suitable because they do not have harsh action, so they do not denature the protein. Therefore, these both type of detergents are used. Example, in case of recovery of cellular proteins in E. coli, Triton X 100, often paired with guanidine HCl, is used for control cell disruption. Third, in case of plasmid DNA, like recombinant plasmid DNA in E. coli, SDS or non-ionic detergents are mainly used for membrane disruption. The plasmid is released out, it is further renatured and purified. Let us understand few advantages and disadvantages of detergent based lysis. First, the advantages: they are very simple and easy to use chemical lysis agents. They are considered mild agents, and thus they are suitable for sensitive fermentation products. Like we saw, they can be used for the recovery of enzymes and proteins. Additional advantage is that no mechanical equipment is required, so this saves the operational cost. However, there are many disadvantages of using detergents. One of the major disadvantages is that detergents contaminates the final product, and therefore removal of detergent during the further downstream purification is very necessary, and it is an additional step. Therefore, due to this additional step, the cost and purification challenges increases, and therefore they are not suitable for very large scale industrial recovery. Also, at high concentrations, detergents can denature the proteins and reduce the product quality. They have limited effectiveness for microorganisms with rigid cell walls. Like in case of gram positive organism, they have very thick peptidoglycan layer which prevents the detergents from coming in contact with their cell membrane. Also, in case of fungal cells, they too have many complex structures in their cell wall which also prevent the detergents from coming in contact with their cell membrane. So because of this property, they are not effectively used against these kinds of organisms. So this was all about detergent based cell lysis. Okay, so let us move on to our second chemical method of cell disruption, alkali lysis. So alkali lysis is a method which uses high pH, that is basic conditions, to break the cells. The common alkalis which are used include sodium hydroxide, that is NaOH, potassium hydroxide, KOH. These strong alkalis causes severe damage to the cell wall and cell membrane of the organisms and ultimately leads to cell rupture. As compared to the detergents, alkali lysis is a harsh method and it is used only when the product can tolerate alkaline conditions. Now let us understand the exact mode of action of alkali. So alkali, like NaOH, when it is introduced, it raises the pH to strongly alkaline levels around 11 to 12.5. At this very high pH, the alkali hydrolyzes the cell walls components, especially the peptidoglycan, by breaking the chemical bonds like peptide bonds that provide rigidity to the cell wall, and this weakens the overall structure of the cell. Further, the cell membrane lipids. In the cell membrane, they undergoes saponification, meaning that the alkali reacts with the membrane lipids and converts them into soap-like molecules, which destroys the lipid bilayer. Due to this, the membrane loses its integrity and can no longer act as a protective barrier. Now the alkali further enters into the cytoplasm and it causes denaturation of the cellular proteins by disrupting the hydrogen bonds and the ionic bonds that maintains the protein's three-dimensional structure. Similarly, the DNA and other nucleic acids are denatured. Because of this, the entire thing that happens, the ultimately the cell membrane and all the cell wall loses their stability and the cell ruptures and all the intracellular components of the cells are released into the surrounding medium. So now let us understand the fermentation products or conditions where this alkali lysis is commonly used. First is, it is very commonly employed for plasmid DNA fermentation. So as we studied in the mode of action, that under alkaline conditions both the chromosomal DNA as well as the plasmid DNA are denatured. However, after the neutralization step, that is when the alkaline conditions are brought back to the normal conditions, this plasmid DNA renatures due to its small circular structure while the chromosomal DNA does not. So because of this property, it is commonly employed for plasmid isolation. Further, alkali lysis is also used in nucleotide fermentations such as inosine monophosphate and guanosine monophosphate. Further, it is also used for fermentation products that remain stable at high pH, like amino acids like glutamic acids, lysine, etc. Now the fermentation products where alkali lysis is not used or should not be used. So it should not be used for enzymes because enzymes lose their biological activity at high pH, and it is not suitable for functional proteins as the structure of the proteins get permanently denatured. Then products like hormones, antibiotics, all these are highly sensitive to alkaline conditions and therefore unsuitable for alkali lysis. Now coming on to some advantages and disadvantages of alkali lysis. First is advantages. It is a very simple and easy method to carry out. It is a low cost method as it uses inexpensive chemicals like NaOH. Then it is effective for bacterial cell disruption. It is a rapid method with very short treatment time, and it is useful for releasing intracellular products, especially the DNA and all the earlier products that we saw in the earlier slides. Now coming on to disadvantages. Harsh alkaline conditions can denature or destroy the sensitive products. Then alkali, as it causes denaturation of many cellular products, so all this leads to higher impurities in the fermentation broth. Also, a neutralization step is an additional step that has to be done in alkali lysis, and it has very limited applications to certain alkali resistant cells or products only. So this was all about alkali lysis. So to conclude, Cell disruption is an essential step in downstream processing for the recovery of intracellular microbial fermentation products. Chemical methods such as detergents and alkali are commonly used depending on the nature of the product and the type of microorganism. Detergents are preferred for mild and controlled disruption of sensitive products, while alkali is used for strong disruption when the product is alkali stable. Thank you for watching this video.